Hi everyone, I am Jean-Christophe Roux from Marseille, and I will talk today about gene therapy, hope, myth, and difficulties. And you will see, I will try to convince you, to explain you why gene therapy is definitively an option to treat or to cure Rett syndrome, but also that we have to face several problems if we want to definitively cure the pathology. Is Rett syndrome reversible? We don't know yet for patients, obviously, but in a mouse model, in this fantastic paper, the team of Adrian Bird, they have shown that if you have a mouse here to synthesize, it's more or less a knockout mouse model. The mouse is really sick and pretty close to die. But if you succeed to activate MSCP2 even late during the pathology, they have shown that there is a strong reversibility of the disease, which is very exciting, and this is why many teams start to work on gene therapy. What is gene therapy? This is an experimental technique that uses gene to treat or prevent disease. And in the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drug and surgery. There are two main types of gene therapy. You have non-viral vectors, and this is liposome and naked DNA. This is lipid plus DNA, but the efficacy to enter in cells is pretty low, and there are some possibility of toxicity. You have also viral vectors like adeno-associated virus or lentivirus. This is virus plus DNA, and this technology helps to enter in the cells to help to transfer the gene with a very high efficacy. But it's possible also to get an immune response. Now, what we know, this is the first difficulty that we have to face. We have to treat Rett girls, and MSCP2, the mutated gene, is located on the X chromosome, and all girls, they have two X chromosomes, but one is more or less disappearing, or it's not working at all. And this is in biology. What we want to do with gene therapy now, this is we want to treat this kind of mosaic of cells. And the good point here to synthesize the red one, this is the sick cells. And if you provide gene therapy, you will probably cure, or you can potentially cure these cells. But if you provide MSCP2 to the good one, the one expressing a normal copy of MSCP2, you will get an overdosage. And what we know that MSCP2 is a tricky protein, because if you have no protein at all in a cell, this is very bad. But if you have too much MSCP2, this is also really bad. And we know that there are some patients, mainly boys, that they have 2 copies or 3 copies, some of them, of MSCP2, and they are in a very bad shape. Now, yes, this is just to synthesize. Here you see in blue the normal cell that is not sick. In pink, this is a sick cells. And if you provide gene therapy, probably we can potentially save the pink one, and the blue one will be sick later on. Now we have another challenge to face. This is how to provide the therapeutic vector into the brain. This is where we want to cure, more or less, to cure the pathology. And of course, we could administrated directly the virus into the brain. And this is one option that some scientists are planning to use. But yes, it's quite complicated because we want to target the whole brain, and this is quite difficult. There are some other options, like injection in the spinal cord, into the spinal cord, and to target the LCR. It means the liquid around the neurons and the brain. Now we have also, if you inject in the blood, this is another option that people are using. This is the option that we choose. But the problem is we have now to cross to the blood-brain barrier. There is a barrier in the brain protecting the brain from virus, big protein, and some bacteria. And this blood-brain barrier don't help very much when you use gene therapy in the blood. But this is a second option. Nevertheless, there are many teams now that they have tried to use this technique, AAV9. This is a serotype of the virus, with MSCP2 to treat MECP2 deficient mice. And the two first here you can see, I put like this. The rescue is pretty strong when you inject directly in the brain, but in newborn mice. This is just before the pathology starts, and this is quite complicated in human beings. There is a weak rescue or partial rescue if you inject in the blood of a young adult mice. And this is exactly what we found in our hand in our laboratory. Now, what this is just to illustrate that one limitation, if you inject the AAV9 vectors with MSCP2 in the blood, here you can see, here the little dot in the control brain, you see many dots. But if you look here, this is MSCP2 provided by the AAV9 vectors, and we got only 8-10% of the neurons that are infected. Another problem, if you inject in the blood, this is what we found in our lab. This is if you inject too much MSCP2, a high dose of virus with MSCP2. You can see in green here, this is liver, the liver. There is a toxicity and there is apoptosis. There is a need then to develop technology to target more the brain and to avoid this liver toxicity. Now there is, just before not finished, but I can show you that gene therapy, there is this nice study that is quite exciting, that has been published several years ago, showing that if you inject not a full size, a full MSCP2 gene, but a truncated gene, a mini gene, it's quite fantastic because since the AAV vectors are quite small, it's sometimes complicated to have a full copy of the gene, MSCP2 gene. But if you use this truncated form, this can help because you can add to the virus some sequence that limit, that reduce the side effects that we saw in the liver. And in this paper, they found it's quite miraculous because they found a durable improvement of mouse model. Survival is improved, breathing is improved, and this is mainly the strategy that people plan to use, the different companies and the different laboratories plan to use for real gene therapy in patients. And now, just before to finish, I just want to mention that there is this idea now. People are probably you heard about this CRISPR-Cas9. This is a sort of molecular scissors, and the idea is to correct directly this is DNA editing, the name. And this is to correct, this technology allowed to correct directly the mutation in situ in the cells. But there allows, there is also there is a limitation, since the scissors are quite big, and the correction, you need to have a sequence of to correct and the mutation, and this is complicated to have both scissors and the corrected sequence in the same virus. But this is something that probably will be a huge option in the future. And now gene therapy in Rett syndrome. Now, as a proof of principle, we have scientists has to demonstrate that gene therapy can cure Rett syndrome in a mouse model. This is what mainly people are doing now. Now, for the safety of gene therapy, we have to determine, to define what is the best option to inject the virus. Is it in the blood? Is it directly in the brain? Is it in the liquid around the brain? We have also to demonstrate that the treatment is safe and well tolerated in big models like primates, pigs, or big animals. We have also to minimize side effects like immune reaction, off-target effect, like we saw in the liver. And we have also to monitor the potential immune reaction in clinical protocol, probably by using also some immunosuppressive treatments, and to use immunosuppressive treatments. And now just I will end by saying thank you very much for your attention. I will pick up your question later on, and you will get a complement of information by Stuart Cobb. He will show you probably some very exciting results in a few hours. Thank you very much.